13. Estuaries are really unique environments. They represent the interface between freshwater and marine ecosystems. And because of this, freshwater flow plays a pivotal role in the nature of these ecosystems. In particular, freshwater flow influences the salinity, hydrological connectivity and productivity of estuaries. It typically carries with it sediments, organic matter and other dissolved materials, as well as biota like phytoplankton and zooplankton into estuaries. And this material, which is typically generated in freshwater or terrestrial environments, can actually be consumed by estuarine biota, and thus it may subsidise food webs and greater estuarine productivity. Nonetheless, estuaries throughout the world are impacted by reductions in freshwater flow from river systems, primarily due to impacts on salinity regimes, connectivity and productivity. Now, the Coorong is one such estuary which is impacted by river regulation, being situated at the end of Australia's longest and most economically important river system, the Murray-Darling. So in the 1930s, a series of barrages were constructed between Lake Alex and the Coorong. These fragmented a formerly contiguous system, and they now act to regulate discharge to the Coorong, which is now on average substantially less than natural. So this change in the flow regime of the region has had some really drastic impacts on salinity and connectivity, and the resulting impacts on biota from these changes are really well understood. What's less well understood, however, is the impacts of this change in the flow regime on productivity of the system. Nonetheless, there's been lots and lots of monitoring programs over the last couple of decades looking at a range of different ecological patterns, and some of which really point at the influence of freshwater flow on productivity in the Coorong. And one such pattern involves this small marine fish called sandy sprat, but it might be a bit more familiar to you as whitebait, and often gets served up in Greek restaurants. This plot here presents abundance of sandy sprat in the Murray Estuary region of the Coorong for about the last decade, and overlaid is freshwater discharge from the barrages. Now, despite being considered a marine species, abundance is orders of magnitude greater during periods of high freshwater flow compared to periods of low freshwater flow. So there appears to be some sort of association here between abundance and freshwater flow. So what do we know about sandy sprat, and how can we potentially explain this association? Like I said, it's a small-bodied marine fish. It only grows to about 70 metres in length. It's common in South Australia in sheltered Gulf environments and also in the Coorong. And in fact, it's the most abundant small-bodied fish in the Murray Estuary or Upper North Lagoon part of the Coorong. It's zooplanktivorous, and itself in turn is preyed upon by a range of piscivorous birds and fishes. Indeed, a recent gut content study showed that it's actually the primary prey item for juvenile mulloway in the Coorong during periods of freshwater discharge. So it's a pretty important fish. Interestingly, at the same time that we found high abundances of sandy sprat in the Coorong with high flow, other monitoring projects also found some other patterns, including really high concentrations of nutrients as well as high abundance of phytoplankton and zooplankton in the Coorong. Indeed, in 2010-11, abundances of zooplankton in the Coorong were orders of magnitude again greater than what they were in the preceding low flow period, including a range of different species that are more common in freshwater environments. So this led us to the hypothesis that maybe organic matter and biota that are being derived in the freshwater environment and being discharged or exported into the Coorong with freshwater flow may indeed be subsidising the diet of sandy sprat, facilitating greater abundance and thus enhancing the greater Coorong food web. Now, this is a hypothesis that isn't new in the trophic ecology of estuarine systems, but it actually has a really well-established basis. And one of the best examples actually comes from the eastern Mediterranean, where there was a large-scale fishery for a species of sardinella, which is actually closely related to sandy sprat. And this fishery was largely focused around the mouth of the Nile River. And the basis of this fishery was annual algal blooms which supported elevated abundance of zooplankton and resulting high abundances of sardinella. However, with the construction of the Aswan Dam and reductions in discharge from the Nile River, these annual algal blooms and zooplankton abundance peaks basically declined, and this coincided with declines in the landing of sardinella and some other commercial species. So the objective of our study was to investigate productivity and trophic subsidy in the Coorong in relation to freshwater flow, using sandy sprat as an indicator by looking at a range of different parameters, including water quality, phytoplankton and zooplankton community structure, the abundance and diet of sandy sprat, as well as the assimilation of riverine organic matter by sandy sprat. And it's these last 2 points that I'll really focus on. So to meet these aims, we used a range of techniques to sample water, zooplankton and fish across 5 sites in the Murray Estuary and 1 site upstream in Lake Alexandrina. And phytoplankton, zooplankton and sandy sprat gut content were investigated using traditional ID and enumeration techniques. We also used eDNA techniques as well, but I'll just focus on the traditional techniques today. And assimilation of organic matter by sandy sprat was done using stable isotope analyses, which I'll just quickly run through. So stable isotope analyses are now commonly used to investigate food webs and trophic dynamics, and actually how they work is quite complex, so I won't go through that in detail, but I'll just talk about how the results of these analyses are interpreted. And it works on the premise that basically you are what you eat. So predators acquire their isotopic signatures from their prey. So nitrogen isotopic signatures tend to become enriched in a predictable fashion from prey to predator, and therefore nitrogen can be used to determine how many steps there are in a food web, or indeed the trophic position of particular species. Carbon, on the other hand, tends to be similar between prey and predators, or slightly enriched in the predator, and thus it can be used to determine potential prey items of predators. But of particular relevance to our study is that carbon that is generated in a freshwater environment tends to be more depleted or more negative in value than carbon that is generated in the marine environment, which tends to be more enriched or positive. And thus, by looking at the carbon isotopic signatures in animals, you can potentially see where that carbon was derived. So we sampled all those different parameters over 3 events in spring-summer 2014, from November through to December. And it's really important to characterise the hydrology of this period. And it was really characterised by a conspicuous flow event in August 2014, which was then followed by low but variable flow of around about 2,000 megalitres a day into the Coorong for the remainder of the study. And prior to that flow peak in August, flow was actually similarly low and variable. To put this in a broader context, this was actually quite a year of lowish flow. Now onto the gut content data. Sandy sprat had a reasonably varied diet. They consumed around about 12 different species of invertebrate. Crustaceans really dominated their diet, particularly benthic harpacticoid copepods, and the next dominant species were cladocerans, amphipods, crabzoea and rotifers. But interestingly, these 2 species, the cladoceran Bosmina meridionalis and the rotifer Keratella australis, both are considered to be truly freshwater species and are more characteristic of environments in Lake Alex or further upstream in the river. This plot's probably a little bit confusing, but it basically presents some more site-specific diet data from sites below Twitchery and Goolwa Barrage. So prey species are across the x-axis, and on the y-axis is Strauss's index of selectivity. And basically what Strauss's index does is it incorporates data on the prevalence of prey in the gut content and also in the ambient environment to tell you whether prey are selected for or selected against. So these green boxes here just indicate instances where these freshwater species I mentioned before were actually selected for by sandy sprat, whilst the blue boxes represent instances where more typically estuarine species like harpacticoids and crabzoea were selected for. And there's 2 things to really take from this, and that's that there are a range of species that sandy sprat prefer to predate upon, but probably of most relevance is that when these freshwater species were present, they were preferentially predated by sandy sprat. Now onto our isotope data. So this biplot presents, again, nitrogen on the y-axis and carbon on the x-axis, and across our trips from trip 1 through to trip 3. And of most interest to us was that the isotopic signatures for carbon for sandy sprat appeared to progressively, albeit slightly, become more enriched across trips. So this would suggest a decreasing influence of freshwater carbon in the isotopic signatures of these fish. This, however, becomes a bit more informative when we look at some past isotopic data that we've got from the species. So we have some data from 2013, which was collected as part of Rhys Johnson's honours project, and that isotopic data actually plots right in the middle of ours. But interestingly, when Rhys collected those samples of sandy sprat, hydrology prior to the collection of those fish was almost identical to that experienced in our study. But perhaps most interestingly, is this data point from Sandy Sprat, which we collected in 2007. Now, this was right in the middle of the Millennium Drought and was at the start of a period of prolonged low discharge. And basically, isotopic signatures of Sandy Sprat from this period were remarkably enriched compared to those from our study and suggested decreasing or even no influence of freshwater carbon in Sandy Sprat in this period. So to conclude, in our study we've shown that Sandy Sprat directly consume freshwater zooplankton amongst more typically estuarine prey species, but that when the freshwater species were present they were selectively predated. Secondly, we showed that freshwater derived carbon is indeed assimilated by Sandy Sprat, and these two lines of evidence together form a substantive base, I think, to suggest that organic matter that is indeed generated in the freshwater environment materially supports productivity within the Coorong, and that this process can actually even occur at low discharges or in years of low flow. The plot on the right here shows annual discharge to the Coorong for about the last 30 years, and the 2014-15 flow years circled here. So as you can see, relative to other years, it's quite a low flow year, but there were still tangible productivity benefits. So lastly, on to management. I think it's really important to note that supporting lower trophic levels or key links in food webs is really important. By supporting these, you do support higher trophic levels as well. And perhaps not coincidentally, but the conditions that support high abundance of Sandy Sprat, high discharge and low salinity in the Coorong also seem to support enhanced recruitment and high abundance of the commercially important mulloway. Now, it's probably no coincidence that these things predate a lot on Sandy Sprat, and no doubt high abundance of a preferred prey item at these times likely contributes to the high abundance of mulloway. Lastly, I just want to say I think this project really presents some more empirical data to support environmental flow delivery to the Coorong, particularly on the basis of productivity benefits. And really this data is vital given the reception to the basin plan initially and continued opposition in some sectors. Such data remains vital. Thank you very much.